So it is an honor for us to have Shlomit Yuval-Greenberg with us today. Shlomit is a friend, and she's now a professor at Tel Aviv University at the School of Psychology. She has done her BSc in Psychology and Computer Science at the Hebrew University, which she then continued to do a master's degree in neuropsychology and a PhD in psychology under the supervision of Professor Leon Duell. She then continued to do a postdoc at NYU in David Heger's lab. Her research involves eye movements, EEG signals. She does a lot of things, mainly with vision, I think, if I'm not mistaken. And we're very glad to have you and hear about your research. So welcome to our seminar. Oh, I forgot to say, she won many prestigious awards and honors. And and welcome to our seminar.
Okay, thank you, Sharon, and thank you so much for inviting me and for holding this amazing series of talks that we're all really enjoying every week. It's really been amazing, and I've been telling everyone about this.
Lovely.
So keep going.
Thank you.
So thank you for introducing me. I'm going to say a few words before I start also about myself, my background, and how I got to study these things that I'm going to talk about today. So it all started in the Hebrew University. I'm assuming you see my presentation, right? Let me know if there's something wrong. And as Sharon mentioned, with Leon Duell, Professor Leon Duell, who was my supervisor on my PhD. And while we thought, we used EEG, this is my son when he was younger, and while we thought we studied gamma activity, we thought we were studying consciousness, object recognition, object familiarity, and other interesting things. What we were actually studying, what we found out toward the end of my PhD, we were actually studying eye movements. We were studying muscle activity, the contraction of muscles that ended up looking like high frequency activity on the EEG. And this was exciting. It was an emotional roller coaster, I should say. This activity was related to microsaccades, these very small saccades that occur around 1 to 3 times per second. And when I finished my PhD, the thing I wanted most was to never hear about microsaccades again, and not to study eye movements, because I was interested in the more interesting organ, the brain. That was why I was there for. So what I decided to do when choosing a place to do my postdoc was to get as far as I can from EEG, because EEG has so many artifacts. So I went on to study fMRI with David Heger in NYU, and they haunted me. Everywhere I looked, there were microsaccades. And this is, for example, a patch of visual cortex locked to the activity, to the onset of microsaccades. And since microsaccades are so affected, and other eye movements are so affected by every cognitive task that you use, especially when you study vision, but as you will see in the rest of my talk, also when you're studying other modalities, everything we tried to do was so much affected by microsaccades that eventually I decided that if you can't fight them, join them. And the rest of my talk is going to be on how to make this bug a feature in my work. So now in my lab, we do eye tracking for almost every experiment that we do. And we combine them with the methods that I was reluctant to use before, EEG, fMRI. We do mobile eye tracking and also desktop stationary eye tracking. So today I'm going to talk about exploration, and specifically about active sensing, and how active sensing is related to eye movements in humans. Let's start with this. Oh, sorry. Let's start with this guy. Oh, what's going on? My video is not playing. Okay. Start with this guy. This guy is exploring. He's using its whiskers to explore. And as you can see, this is what active sensing is all about. So this rat is not just perceiving. He's perceiving and acting upon his perception, its perception. So, so this is what exploration is all about. It's not only perception, it's more than that, it's exploration. There is a combination, interaction between vision or perception and motor activity. Feel free to stop me for questions if you have any. So this is also what we do. These are not us, but we can look at this image with the cuts. We human, we use mainly eye movements to explore. This is our main method of exploration, visual exploration. And what you see here on this image of parrots is are our fixations. When we look at this image, this is a typical subject looking at this image. We focus on places of interest. And this exploration is such a strong drive, even an automatic drive, that as my student Dekel, I'm sorry, before that, we do saccades around 1 to 3 times per second. Even if we are asked to fixate, if you give this participant this fixation cross and you ask them to fixate, they still do saccades, they do micro saccades, these very small saccades, and they still do them around 1 to 2 times per second. So we can't help it. We want to explore, we want to move our eyes, we want to learn. Even if we get this task to fixate, there is something going on and exploring. And now we get to Dekel. As Dekel, my unfortunately ex-student, showed in the lab, this exploration is so strong, it's such a basic human drive that even if we ask participants to do a task that completely doesn't require exploration, such as in our case, it was mathematic computation, like we gave them arithmetic questions, still while they do it, they explore and they visually explore images, and they do so very, very similarly to the way they would explore when given a visual task. So visual exploration is a very strong drive. We almost can't help it, even if it doesn't help our task. Sometimes even when it interferes with our task, we still explore. So the question that I'm going to ask in this talk is, when do we stop exploring? So what are the functions or the mechanisms that are so strong and so important to us that they will end up with stopping, their consequence would be to stop exploration, to affect exploration, to slow it down, or to completely stop it for a short time. And I'm going to give you a spoiler at this stage. The answer to this is expectations. So what we're going to talk about today is how expectations, both in time and in space, affect exploration, interfere with exploration, or change exploration. And it's kind of trivial that that expectation would change the way we explore the world. But I think one thing that I'm going to talk about today, and I think that was neglected in this literature, even if it's, when you think about it, it's kind of trivial, is that in addition to active sensing, like to this rat going forward and looking for things that are interesting or important, there is also the opposite side of active sensing, and that is, I call it active avoidance. So sometimes the right thing to, when you're an active perceiver, is not to go ahead and explore, but to avoid, for example, when there's a distractor. And this is an aspect of active sensing that is rarely mentioned or studied. And this is what part of my talk is going to be about today. So to outline my talk, I'm gonna talk about 3 loosely connected projects. The first one is going to be about how spatial expectation affect your gaze, or more specifically, your gaze aversion. This is exactly what I just talked about, how you do this active avoidance when you need to. Second project is about temporal expectation. And the third project that I'm going to talk about is about temporal, if we have time, is about the temporal dynamics of eye movements during exploration. So let's start with the first study, and we're going to start with a very short demo. or exercise. And this works 100% when I'm in class. I'm not sure if it would work on Zoom, but please try to work with me here. Try to calculate this. Do 35 times 12. Please do it, really, for real. Okay, I'm looking at your faces now. It works with some of you, but not with all of you. What happens when I'm in front of people physically, that more or less 100% of the people will look up to the ceiling when they do that. Or some of them look on the floor, but none of them look straight ahead of me. And in fact, when you write thinking in Google and you look for images of thinking people, this is what you get. So when you think, you look at the ceiling, you look up. Why do you do that? There are many answers that can be given. This is in fact the question that that drove my student Dekel to his PhD. That was his question behind his 5 years of research. But this led us to many questions, understanding that this is a complicated, there are complicated answers. One of them is social embarrassment. So we look up, and this is why it doesn't work so well with Zoom, because I'm not there and you're not so embarrassed. But there's also another aspect, and that is a more perceptual aspect. This is a method for avoiding distraction. And we showed this in a simple experiment when we presented participants with this grading that was moving. It was either moving very fast or slower, which was either being a very salient visual stimulus or less salient visual stimulus. We presented participants with a number, and they were asked to then calculate arithmetic question that was presented to them auditorily to not affect their eye movements. And while they were presented with this question, we monitored their eye movements. They were calculating. At the end, they just had to indicate whether the answer was the one that they was bigger or smaller than the one that they were presented before, within the number that they were presented with. And what we found, we looked at gaze aversions, how much people spend outside the grating, how much they averted their gaze. And we found that they avert their gaze more when they are presented with a fast stimulus relative to the slow stimulus. So these are individual participants. Everyone that is above the line averted their gaze more for fast stimuli, i.e. salient stimuli, than for the slow stimuli, and opposite for these ones. So you can see that almost all the people, all the participants showed this effect. So salience, visual saliency affect your gaze aversion. You tend to avert your gaze more when the stimulus is more salient, indicating that probably this behavior is a way to avoid distraction. And this is basically the first demonstration for this active avoidance. You're actually moving away from this distraction. And feel free to ask me questions if you have. Just unmute yourself and burst in. But the next question that was more and more important to actually show that this is part of an active sensing mechanism is to show that this behavior is dynamic and predictive, to show that this is something that changes, that can be modulated by changing stimulation. In other words, it is actually active. So with his next project, Dekel did something a little bit more complicated. Participants were presented at the beginning of each trial with a number. Then there was a fixation. And I'm sorry, this is not fixation. This is a mathematic operator. In this case, it's plus, but it could also be minus. And their task was to add or subtract 7. So after you get this 172, you were supposed to add 7 at each stage. Just do it at your own pace. Every time you add 7, you press a button. You don't say the answer aloud, you just press the button and you remember, and you keep going, either adding or subtracting. And while you were doing this, you had a visual, we call it distractor, although it was completely task related. So it was not really a traditional normal distractor. There were visual there was visual stimulation on the screen. In this case, it was numbers to make it more distracting, but participants were not told to do anything to do with vision. Everything was supposed to be the arithmetic calculation. And this distractor always appeared in one corner of the screen for the entire block. So it was 100% spatially predictable and temporally unpredictable. So they never know when it's going to come, but they know for sure after seeing the first time that it's going to appear in this case, left top corner, and in each block it was in a different corner. And we look at the gaze, and we look at the gaze only when there is no stimulus on the screen, because we are interested in prediction. We're not interested to see what are the responses of the eye movements to an actual physical stimulus. We're actually interested in what happens when there is no stimulus, when the screen is empty and they're just preparing for something that is supposed to occur. And this is the result. You see here, X and Y represent basically locations on the screens. This is normalized, so it's relative center of mass. I'm not going to get into the details of how this was calculated, but for us, it's enough to understand that this represents the screen. This corner represents the corner that was distracting in this specific block, and gaze was averted away. It's always to the opposite, to the diagonal quadrant. This was the result for when the distractor was on the top right, bottom right, and bottom left. So it's always to the diagonal quadrant. Notice something interesting, which was surprising for us. This is not a huge, to say the least. It's a very small aversion. So they didn't actually shifted their eyes to the ceiling or to the floor as we would do when asked you to calculate an arithmetic exercise. This is interesting. It's just a small shift, but probably is enough to make this stimulus a little bit less distracting. And we will talk about this in a few minutes again. So in this study, gaze was shifted away from the predicted location of an irrelevant visual stimulus, taking this active avoidance to another step, saying that this is a predictive active avoidance, more connected to active sensing and to what we see with other active sensing mechanisms. And one way that is interesting to think about this, or to explain it, and to put it in a context of some kind of theoretical model that we try to do, is when we think about it in terms of relevant saliency maps and relevancy maps. And I'm going to spend 2 minutes explaining these two terms. Let's say you have this stimulus, you have two stimuli. One is high contrast, one is low contrast, but one is a target. And the low contrast one is a target. So we have contradictory purposes here. We have the saliency and we have the attentional priorities, and they're distributed differently in space. So if I have a neuron that is part of a saliency map, this neuron would respond to saliency. So it would respond to the white circle over here. But if I have a neuron that is part of a relevancy map, more affected by my attentional priorities, it would respond to the target more than to saliency. And these are two separate concepts in the brain. Well, what we suggest with the model, with our theoretical approach, is that this what we call predictive active avoidance could bridge these two maps. And I'll explain how. Let's talk about this distractor that we showed our participants. Let's say, for instance, that it was unexpected, I mean spatially, which it actually was, but just for the sake of explaining, let's assume that it was unexpected. What happens? We fixate in the center, more or less. Looking at our saliency map is very affected by fovea. So nothing at this stage happens on the screen. The screen is blank. So it should be like a unified saliency map, nothing affected, but still our fovea is the area that we are more tuned to. So our saliency maps look something like this, giving priority to the center. How should our relevancy map look? Nothing's going on because right now we have no task and no expectations. And then the distractor comes on, and distractor shifts the saliency maps. So this is salient, everything else is unsalient. So we're getting this, imbalanced map, and a relevancy map is unchanged. But when the distractor is expected, like in the actual experiment that we did, and we fixate on a blank screen, our saliency map is still the same. It's not affected by expectations, but our relevancy map is affected by our expectation. Yellow is areas that we think are relevant or that we prioritize. In this case, this is a distractor. It's not the target. So everything is strong except the area where the distractor is, because we want to prioritize perceiving everything else. We want to avoid the distractor. It's unhelpful. And the outcome of here is a shift of the gaze, because when you combine these maps, you get the map that drives your eye movements. And with these two maps, the saliency map that is completely balanced and symmetrical, and the relevancy map that is now asymmetrical, the result is an eye movement, and the eye movements would be away from the distractor. So now the important part of the deal is that we change the saliency map. By shifting the gaze away, shifting the fovea away from the distractor, you actually made the distractor less salient. So you made it less effective. And this would be a very efficient way to handle distractions. And again, I think when thinking about it, okay, we move away for something distracting. This is it's very trivial, and it really is very trivial and simple. The thing is that if you look at literature on distraction and how the cognitive system handles distraction in the visual system, there is almost none or even no research on how you actually physically avoid the distraction. There's a lot on how you suppress them. Participants are usually asked to fixate. They're never allowed to move their eyes, as we did in our test. When they're asked to fixate, true, there are brain mechanisms that work through perception to suppress activity in the areas that are distracted. But what we want to say is that you can't detach the motor aspect from the perceptual aspect. If you let participants just move their eyes, you see that a huge part of avoiding distraction is active avoiding it. Moving away, reducing the saliency, affecting not only your priority, but also the actual stimulus that you see by making it more peripheral. So to summarize this project, and then I'm going to open up for questions, is visual exploration can be flexibly modulated by spatial predictions. Suppressing distraction involves physical avoidance and not only perceptual suppression, as most literature in this field show. And active avoidance can be a link between saliency and attentional priorities. Questions so far before I go to the next project?
Shlomit, there are two questions in the chat if you want, where I can read them to you.
Better read, because for some reason I can't find it. Oh, here it is. I found it.
Well, I can ask mine.
Oh, did they also close their eyes? Nama, what is I'm not sure which part of my.
I think it was about.
Can you hear me?
Yeah.
Yeah, another way of avoidance is not to shift your eyes, but just to close your eyes. So I was wondering if you tested whether they closed their eyes.
Yeah, of course we did. By the way, it's difficult for us with our eye tracking, with the way we did this experiment, and we're thinking about how to change this. I would be really happy to hear suggestions, but with our eye tracking, it's very hard to dissociate between closing the eyes and losing or moving outside the trackable zone. So what we see is nuns. But when we look at our participants, it doesn't look like they close their eyes for long intervals, more than blinks. They do blink more than more when you give them more salient stimuli. And this is probably part of avoidance, but interestingly, it doesn't look like closing your eyes is a method that is used to avoid distraction, although it should be a more efficient method than shifting your eyes. And we think, this is just open questions, we haven't checked that, but we think that this is related to survival. So we don't really want to close our eyes. This would disconnect us from a world where we supposedly could be attacked by a tiger, or I don't know what, maybe activate some brain activity related to sleep or resting. We know that there's alpha activity when we close our eyes. So there could be reasons why we don't do this, but as far as we see, this is not a method that is used.
Thanks.
Does the subject know his gaze shift from the visual distractor? We asked them, no, they don't know. They don't know. They also don't understand the point of the experiment, which is important to us because they don't do anything. Nothing of what they did is explicit or aware, or they don't. Of course, when you tell them, when I told you that you're looking to the ceiling, you were aware that you did that. But the fact is that our participants, we asked them, they can't explain what happened. Will the visual distractor enter working memory and occupy resources of working memory? I don't know. I assume that if it's not foveated, then we don't see much, especially that it's numbers. But I can't tell more than that because we never asked anything about the distractor and purpose. We wanted the task to be completely non-visual. And this is like a crucial part of our procedures. That's why we don't know how and whether the distractor was processed.
Shlomit, I would also like to ask, first of all, it's beautiful. I want to ask if you would change the distractor to a different type of visual stimuli that is not related to numbers, for example, pictures or even words, or just letters, do you think that there would be a different avoidance pattern?
Yeah, that's a good question. What we did in our experiment is to use numbers, as I showed you, but there was also inverse numbers because we wanted to ask this question. I didn't show because it was completely identical. No difference at all.
So I want to continue on this question. So can you change the degree of avoidance by changing the saliency of the stimulus, let's say contrast or something like that?
This is a good question, and I wanna check that because I think that the reason why we saw, I don't have an answer yet, in other words, but I think the reason we saw such tiny aversions, this was very surprising for us. I don't know if you noticed, but it's less than 1 degree. With the type of stimuli that we used, that makes sense because with numbers, when you shift 1 degree from the fovea, it's much less recognizable and much less interfering. And one thing that I want to do is, for example, just to enlarge them and see if we then enlarge also the aversions. So I don't know.
You did ask to fixate, right? They had to fixate for to read the.
So yeah. So we did start each trial with number at the beginning in the center. They had to see this number and the operator that came after it. But then the trial was long. It was, I don't remember, but like 20 seconds or something like that. It was long. And very importantly, we never asked them to do anything with their eyes because this is what makes this procedure different than other studies on distraction when they ask people to fixate. So we don't tell them anything. I don't remember the details, but Dekel told them something about eye tracking or why we do that, to kind of not focus them on, it's not a study on eye movement. They didn't get that at all. So as much as we could, we never said anything about eye movement. So they could close their eyes, they could look at the ceiling for the entire trial, but they didn't do that.
Well, they are probably trained subjects for other experiments or something.
They were, most of the subjects were first year psychology students. They did other experiments, that's true. So this is why we also asked them at the end. None of them felt that they were obliged to do something, like to look at something but yeah, this is a possibility. We didn't use naive subjects.
What about
We're
manipulating
not lab
relevant?
members, I should say. We on purpose for this studies, we don't use lab members, at least that.
What about manipulating relevance? So if you write 172 in the top left corner.
Again, something that could be interesting and we never tested. What we did, I should say, if like just that manipulation that for lack of time, I didn't mention here, is that we did a replication experiment and we added like anchors, like just 4 dots on the screen on the 4 quadrants, just so that we thought maybe we could give them like something to look at, if that might change. And actually, I should mention something interesting here, which I wasn't planning to talk about, but since this is a vision audience, I should say that there was something that that was important here and kind of stupid. But after we did our first experiment, the one that I presented, we looked at the map of gaze and we saw that during this entire time when they were not supposed to look at anything because everything was blank, a lot of them tended to focus on the left bottom corner of the screen. And this was on top of our effect. So the effect was there after normalizing. We did something like baseline correction. And then we found that the shifts were still there. But if we look across the entire trial, we see this shift left and bottom. And it was like we've gone mad to try to find out why this is so, why left. Eventually, after a lot of work, we found that there was a tiny I don't know how it's called, like a perul, like of dust particle on the screen. It was so small you could not see it if there was anything on the screen, but only with a blank screen and people that were looking for a long time, you could see it. And this was the reason. So this is what brought us to think that something about gaze is, so you want to look at something, and this is a very strong drive. And I think this is very important for people studying vision to know, because since then we actually physically wipe our screens after every subject, so that at least we don't get anything consistent if we do have still some dust in the air. And this is why we ran the second experiment as a replication, and also to test what happens when you give them these dust particles, like actual dots, and there was no difference. So it had no effect at the end. So this is kind of diverting me. There are a few more questions on the chat, I think, but maybe should I move to the next project and then go back to
I think that, yeah, great.
Okay. So the next project is on temporal expectation. Now so far this was all about spatial, and now we're going to talk a bit more about temporal expectations and how they are affected or how they affect eye movements. So what are temporal expectations? Temporal expectation is simply the ability to extract temporal regularities and act upon them to improve your perception of predictable events. So let's say, for example, that I'm waiting for a bus. There is some regularity, at least hopefully, with the time buses arrive. And this regularity could take a few different forms. For example, it could be based on rhythms. For example, if I live, I guess, in Europe, in Switzerland, then there's a timetable. And the bus comes, for example, every 10 minutes, and there's a rhythm. So I know that every 10 minutes, there would be a stimulus, there would be an event, a bus. But I live in Tel Aviv, and there is no regularity. There is actually something, because there are these digital boards that tell you that it would come in 2-3 minutes. So this is kind of like a cue or an association-based expectation. So if I see this cue, the bus will arrive in 2 minutes, I know that it will probably arrive in 2 minutes. So this kind of temporal expectation is a cue followed by a stimulus with regular intervals between the cue and the stimulus. This interval between the cues and the stimulus is often called foreperiod. And last, there are many others, but these are the 3 that I'm going to talk about today. hazard-based expectation. These are expectations that are based on conditional probabilities, on the idea that if something has not occurred yet, what is the probability that it will occur now? So this is someone now waiting for the bus for a long time. It's already night. She looks a bit depressed by this long wait, and but she knows that because the bus is bound to arrive at the end, then the passage of time increases the conditional probability that it would arrive. So if it had not arrived by now, it should arrive with higher and higher probability in the next moment. So this is called hazard-based expectations. How do you measure temporal expectations? So most of the studies so far use behavioral, traditional behavioral measurements such as reaction times, accuracy rates. Some other studies, and I'm talking studies in humans, they use brain responses. The problem, there are different problems, although these methods are useful, there are different problems with them. The problem with behavioral metrics is mainly that they are retrospective. So I'm actually measuring the consequence of expecting and not the actual expecting. So expectation is something that comes before the event. And when I'm measuring behavior, I'm actually measuring their consequence. The problem with brain responses, especially with humans, is that they are difficult to interpret. So there are a lot of studies with EEG, for example, on temporal expectations showing, for example, the CNV component, and it's very difficult to say what exactly it reflects, how we can interpret it. So what we have been doing in the lab for the past few years is to use eye movements to study temporal expectations. And we think, and we show already, that they are potentially good candidates as indices for temporal expectation, and also as part of understanding these mechanisms. So these series of small projects are asking more general questions of, are eye movements affected by temporal expectations, A, and can eye movements be used as an index to study these various types of temporal expectation that we see? Why did we, at the beginning of this why did we think that eye movements should be interesting in this respect? First of all, because eye movements are continuous. So more similar to maybe EEG when we measure it, we can measure eye movements at any time. People do eye movements all the time, as I said, like 1 to 3 times per second. Therefore, we don't have to ask the participants to do anything in order to produce the eye movements. They just occur all the time. So they're continuous, and they provide temporal information even before the event. Also, they're implicit. Again, we don't have to ask participants to actually perform a temporal expectation task. We don't need instruction, we don't need cooperation, and this would help us use this index as in the future to study temporal expectations in populations that are less cooperative. For example, we're now about to start using this method to study temporal expectations in infants. And finally, this is a behavior. After all, this is behavior. It's not brain. So it's supposedly much easier to explain. We will see that this is not as simple as we originally thought when we started this work. So we started this line of work using looking at rhythmic temporal expectations, the work of Yarden Dankner, who did that for her master, and now she's already a PhD student. And we used a task that is often used as a diagnosis task for ADHD. It's called the CCPT task. It's a very easy task. The task is just to detect a target, a red square, among other geometrical and colorful objects. And this is a rhythmic task, which could, we contrasted the condition when these objects came rhythmically with the condition when they were non-rhythmic. So they came at random intervals. And we looked at eye movements. Now, y-axis is saccade rate, how many saccades we see. Most of these saccades were microsaccades, small saccades. but we didn't give any instruction with regards to eye movements. So some of them were larger. X-axis is time relative to the target. 0 is the target, the stimulus, this object. And what we see here is saccade rate in red. This is in the non-rhythmic condition. In blue, it is saccade rate in the rhythmic condition. We are interested in this 100 milliseconds right before the target. This is something else going on here. We're not going to talk about this today. We're focusing on the period of time just before the stimulus and comparing rhythmic and non-rhythmic. And what we find is that saccade rate is decreased for the rhythmic condition relative to the non-rhythmic condition, which could be suggested that this could be used as an index to study temporal expectations using eye movements. By the way, unrelated to the topic of my talk today, but I should mention it, in this study, we, which I did together, I should say, with Lilach Shalev from our education school and with Marisa Carrasco from NYU. This study was actually about ADHD. So what I show you here is the neurotypical group, those that were not diagnosed with ADHD. And when we look at people that are diagnosed with ADHD, there is almost no effect. And what I don't have here today, but should believe me, when we looked at people that have low abilities to sustain attention, even those that were not diagnosed with ADHD, when we take our entire pool and divide it to those that are high and those that are low in sustained attention, this effect is completely gone. So we found some very strong link between this effect and sustained attention. And this is one example why this is a good measurement, because we can use it with this special population very easily. Someone unmuted for a question? Okay. I'm not following the chat, so please just unmute if you want to stop me on for something here.
Okay.
And also, Sharon, stop me for time if I'm not following time. So this part, the neurotypical part, indicates that what we call the pre-stimulus or pre-target oculomotor inhibition could be used as an index for rhythmic-based expectations. But then there was an open question. What about other types of temporal expectations? For example, cue-based, association-based. My also ex-student, Amit, asked this question by using a condition when there was a fixed interval or a 4 period between the cue and the index, and the target, and a condition with random. It wasn't completely random. It was one out of 5 options, intervals between the cue and the target. So this is basically asking the same question, but this time we used a more psychophysical task with a cue, a fixation that changed color, and this color change indicated that our target, which was this rating, will occur in one of 5 4 periods, 1 of 5 intervals. And in one condition, this was fixed for the entire block. So it was always 1, for example, or always 1.5. And in another condition, it was random, 1 out of these 5 intervals. And then participants were asked to indicate the tilt, the direction of tilt, whether it was clockwise or counterclockwise. So this was done in separate blocks, fixed versus random. Okay. Again, y-axis is saccade rate, x-axis is time, 0 is the time of the target. And for this, I'm presenting only one specific 4 periods, the 1 second. I could show others. This is the 1 second 4 period. And when you compare fixed and random, we see exactly the same thing, the same effect that we saw with rhythmic expectations. So the fixed goes down here, randoms is up, and when we focus on the last 100 milliseconds before the target, we see this very large effect, much lower number of seconds for the fixed relative to the random. So it looks like pre-target oculomotor inhibition is an index also for association-based expectations. And we see something else that is interesting here. If now we look on all the 4 periods. 4 periods in the last slide, I showed you only 1 second, but if I show you a bar graph with all of them, you see this very large effect between, this is mean saccade rate for the last 100 milliseconds before the target. You see this largest effect that we saw in the previous slide for the 1 second, 4 periods. It was very significant. We still see a large effect for 1.5. For 2, there is no effect. For 2.5 seconds, there is a slightly significant effect, but it's small, and then no effect for 3 seconds. So when we increase the time between the cue and the target, this 4 period, the effect goes down. And why does it go down? There are 2 reasons, which I think you can see from this graph. Let's look for a second only on the fixed. You see that it goes up. So when the interval between the cue and the target goes up, expectation is reduced, meaning that saccade rate goes up. And this makes a lot of sense. We see the same effect with reaction times, because we know that it's much harder to estimate the duration of times when time is longer. So for 3 seconds, it's already difficult to estimate, and therefore your temporal expectation or any effect that is related to temporal expectation would be smaller. So this inhibition is smaller. Remember, this is inhibition. So smaller inhibition would be larger number of saccades. And now let's look for a second on the random condition, on when intervals were unexpected, and we see the opposite effect. So with time, saccade inhibition is stronger, saccade rate is lower. Why does this happen? This was also expected, and this is also something that we see with reaction time. This is the hazard rate effect. Remember this person waiting for a long time at the bus stop. Now she's waiting for a long time, and when time goes by, her expectation grows. And when we are at this random condition, it's exactly like her waiting for the bus. So with the 4 period increasing, our inhibition increases also, and saccade rate goes down. So this is an indication. It's not a proof yet. I left the question mark here because we need some more evidence for this, but it looks like the pre-target oculomotor inhibition is also affected by hazard-based expectations. So to summarize until here, we saw that if we look at this oculomotor inhibition effect, it's related to rhythm-based expectation, it's related to association-based expectation, it's most probably related also to hazard-based expectation. But why? So this is the big question that we're still struggling with. We don't have an answer. And if to connect it to the first part of my talk, the way I think about this is that there is some inhibition of exploration here. So we know something expected, and you reduce your exploration before. Remember, exploration is a very automatic, very strong human drive. We want to explore, but still, when something is temporally predictable, when there is an event, we know that we have to respond to it, exploration goes down. Why does it happen? So there could be a trivial answer to this. These were visual stimuli. So you would want to avoid missing them. So a trivial answer would be we don't do eye movements before a stimulus appears, because then we might miss the stimulus. So the question that we asked is this oculomotor inhibition simply a way to avoid making a saccade right on the stimulus? And there could be different ways to answer this question, but one thing that we did together, Dekel did together with Ro'i is to use a stimulus that is not visual, simply to look if we find this effect when there is nothing visual going on the screen. And to do that, we based our procedure on this exactly the same procedure that we use with visual stimuli, only everything visual was replaced. Both the cue was now auditory, it was like a tone, and the target was auditory, it was pitch discrimination, like a little bloop, that participants were asked to discriminate whether it's going up or down. again, same 4 periods, same temporal regularities. Comparing random and fixed here with the 1 second 4 period, you can see here in the last 100 milliseconds, we see a similar effect. It's smaller than with visual, but it was still highly significant and very consistent across participants. If we now look separately on the 5-4 periods, we don't see as clear this reduction and going down and going up as I showed you before, but we still have significant effect for 1 and 1.5 4 periods. So there is something, this oculomotor inhibition exists, and it's not visually specific. And this was a very important step toward explaining why is this mechanism, where does it come from in the brain? We still don't have final answers for this, but we know that probably oculomotor inhibition is part of a mechanism of temporal expectation that is modality free, that is supramodal, that is unrelated to a specific modality. After we did this study, I did together with my collaborator Marisa Carrasco another study showing the same effect in the tactile modality. So again, we know that this is not specific for vision. Last, before finishing describing this project, I would mention a study that we are currently doing together with Noam Tal-Peri, who I see is here in the audience. And what Noam did is to ask whether we see this effect in free view. And this is a very important question, first methodologically, because if we see it in free view, it would mean that we can go the next step toward using it as an index with various populations. Because, for example, with the infants that we're going to soon start working with, we can't ask them to fixate. And second, for me, at least, what this was important for is to generalize this effect and to show that it actually related to expiration. So with all the previous studies, we didn't ask participants to fixate, but there was a fixation cross on the screen. As mentioned before, most of them did experiments before and they were thought they were asked to fixate. In this experiment, we did exactly as the auditory experiment that you saw before, but there was an image and there was no fixation cross, and they were not asked to do anything with their eyes. And still, interestingly, we find, as you can see here, the yellow and the purple, we find the same effect coming before the target. This is the last 100 milliseconds before the target, and we see a huge effect, and the effect is actually identical if you fixate or don't fixate, which is, again, very interesting because it tells us something about microsaccades, about these small saccades that occur during fixation, and that their role, at least in this context, is very similar to the large saccades that we do while exploring. So why are saccades reduced prior to an anticipated visual target? Well, the interpretation that we now give is that it's related to exploration. So it is not only saccades that are modulated by temporal exploration, but also by temporal expectation, but it's actually exploration that is modulated by temporal expectation. So very similar to what I called the active avoidance effect that we saw when we gave the participants something that is spatially predictable. Now when there's something that is temporally predictable, they inhibit their exploration, they wait and prepare to receive this predictable stimulus. So the strong basic human drive to explore can be inhibited when you're expecting something. So to summarize this, oculomotor inhibition is a reliable index for multimodal, not only vision, temporal expectations of various types. Exploration is paused in anticipation for predictable and task-relevant events. And I skipped. No, that's it. So questions on this part of my talk. And I'm going to try to find again the chat that disappears. Is the reduction of exploration linked to learning mechanism taking effect, e.g. needing a verification of the expectation as a way to learn and update our expectations? So every expectation is in some way related to learning, because we need to learn the regularity. If there is no, if we don't know the regularity, we can't do any mechanism that is related to expectation. We think, for example, that this is what happened with the ADHD, that the problem was not with actually forming expectation, but specifically with this task that that challenged their sustained attention and made it very difficult for them to learn the regularity. But more than that, I don't know yet, because we haven't tested this question directly. Another question, is there any connection to the reducing of the microsaccades to the phenomenon of saccadic inhibition? Is there any connection between the reduction of the microsaccade to the phenomenon of saccadic inhibition? I'm assuming that the question refers to the post-stimulus saccadic inhibition. We see some balance. We see sometimes that when there is stronger inhibition, so saccades need to come out sometime. If they're inhibited before the target, they will be more likely to occur after the target. So we see that after, in one of my images, you saw that there's inhibition and then this large rebound coming after a stimulus. And we do see that it's a lot of times correlated with what happens before the target. So when there's stronger inhibition before the target, the post-target inhibition would usually be smaller because you can't inhibit saccades for too long. More than that, again, I don't know, because we haven't directly tested it. We just looked at we always focus on the time before the target to see how it's affected. There are a lot of studies, for example, by Yoram Bonneus here, showing that this similar kind of things affect the post-stimulus saccade rebound, inhibition, and then rebound, and prediction and expectations were shown to affect this part of the dynamics as well. So I think both parts of this are very connected, but we haven't really characterized this connection yet. Another question from the chat. I guess 4-period inhibition of saccade is determined by duration of dopamine secretion and recycling. Am I wrong? So this is a big question. I'm always, when I'm writing these papers of these series of projects, we're always debating whether we should suggest anything to do with the brain or not. And we have a series of candidates for the brain mechanisms that we think are involved. And it's very, very difficult to answer this question using experiments on the brain. We haven't actually figured it out yet, because there are a lot of things that are related to each other, like very difficult to manipulate eye movements and also hold the temporal expectation controlled, or doing the opposite. Everything affects everything. Very, very hard to dissociate in a brain experiment. But yes, dopamine is one of the leading candidates, and it's very likely part of the story. But there are also other candidates. Can you link behavior to the eye movements, e.g. when you move away your eyes, you perform better? Sorry, is this related, sorry, to the previous study or to this one? Probably to the previous one. Okay, I'll answer it anyway. Again, it's part of the same answer as on the brain mechanisms, because it's very hard to dissociate. Because when you move your eyes, you're performing, it improves when you move your eyes from a distractor, it improves your performance, because it's less than a distractor. So it's very hard to control for how these two effects, like performance and eye movements, interact between each other. But yes, I think the answer is from what we see. So what I mean to say is that we haven't actually did an experiment when we asked people to look on the stimulus or look away. But from what we see is, yes, when you avert, you perform better. But we don't know, for example, if if you perform better, because these were the trials when you were more attentive to the stimulus, and this is why you both moved your eyes away and performed better. But the short answer after all this is, yeah, that I think that it's very linked, and I think this is the reason why you shift your eyes away. The question is whether, I think I should probably not move to the third project. Sharon, you tell me.
Well, I think perhaps if you have just, I mean, if you can say just, very concisely in a few words about what you did and what the results are, that's fine. But if it's, if it's a elaborate description, then maybe we will leave it for your next talk.
Yeah, I think it would be a little, with that project specifically, it would be difficult to explain in brief. I regret that I didn't put it before, because there I talk a lot about Ayelet's study. If I knew that Ayelet is going to attend, then I would have put it earlier. But I think, yeah, it's better to wait for next time with this, because it's a lot. So maybe I should answer more questions from the chat. Oh, I think I answered all of them.
So, so you want to wrap up before we give you a big applaud, or?
Sure. Let's keep everything so that you can wait.
Well, the slides look lovely.
Yeah, this is Ayelet's study, so I gave.
Okay, now we're intrigued. So maybe Ayelet can tell us about this when she's giving her talk. No, not.
I recommend. Okay, so I'll just give you the general conclusion. So we talked a lot about visual exploration, and the main take-home message here is that visual exploration through eye movements is a dynamic process. That's the most important thing. It's a dynamic process because this is the part that I didn't have time to, but it is a self-paced loop of perception and action. So action affects perception, perception affects action. As we saw with this mouse, with this rat, we can't dissociate these two things because they each affect each other. It is modulated by expectation, both temporally and spatially, as we saw before, and it's modulated by task demands. And I think these entire group of findings, and in general, the study of eye movements together with vision demonstrate how important it is to get a comprehensive view of vision as part of this active vision, part of this system that combines motor and motion and perception together, which you must study together in order to understand the complete picture of perception, attention, and motion. And with that, I would like to thank the 3 PIs that work with me on these projects, Marisa Carrasco, Lilach Shalev, and Izar Bargad, and also the students, Yarden Dankner, Dekela Beles, Noam Talperi, and Roy Amit. And thank you, and thanks Sharon for inviting me.
Thank you very much. I'd like to invite everybody to unmute themselves, and let's give a big applaud to Shlomo.
Thank you.
Thank you.
So, wonderful talk, very inspiring and beautiful. And yeah, I'm inviting everybody to unmute themselves and ask questions if that's okay with you.
Sure.
Can I ask, what do you think? Does this tell us anything about the role of microsaccades? That is, they show your effects, presumably because of this blanket of inhibition that is brought down. But do you think it tells us anything about why we're making the microsaccades?
Yeah, so I think that this was not the purpose of these studies, although I think what we do see, and we constantly see with all our studies, not only these, is that microsaccades are just like large saccades, in the sense that they are a part of exploration in every sense. So when in some of these studies, as I showed, when you put this fixation cross on and you ask participants to look at it, you basically get a minimized version of the same thing that you would get if you would not ask to do anything specific with the eyes and just let them free view. So what I think, and this is one of the views that people say today, is that microsaccades are part of the exploration system, probably tuned to explore finer details and smaller items. And when we ask participants in the lab to fixate, we target something that is not a naturalistic behavior, and we activate this mini exploration system just on a small scale. But in my hand, in my years of studying eye movements, I've never encountered any inherent difference between microsaccades and larger saccades, except their sizes, of course.
I mean, it could be just to prevent some kind of refractive response from individual cells. I mean, you know, when you have a stabilized image, the stimulus fades. And I mean, it could be a mini version of that you'll get.
You mean to avoid fading?
Yeah.
Yeah, so I think it's part of the story. I think that if you look about evolution and why microsaccades developed, it's maybe the search for a purpose that in the last 2 decades, probably, of this literature, there was a lot of debate and arguments on what is the purpose. That we don't have, there shouldn't be one. Of course, everything that is that has some advantages should survive. And I think that avoiding fading is one aspect of microsaccades and why they are helpful, and why maybe another reason why they survived evolution. But avoiding fading could have been done also by head movements or body movements. So it's not, I think it's not everything. And also we find that they're not random as people thought when they started studying microsaccades. We see that they're very related to every cognitive task that we give participants, affect their eye movements, either their direction or their rate. And what is, again, more important is that the way these cognitive manipulations affect microsaccades is exactly the same as it would affect larger saccades during exploration. So I think being part of exploration is what microsaccades are all about. But yes, probably they have other evolutionary advantages, such as avoiding fading.
Can I ask a question? Okay, first, very nice talk, Shlomit, and nice work. And I wonder if you think that the same explanation, so you argue that you stop exploration when you are anticipating something to happen, right? That's the...
I don't want to stop, you inhibit. You
inhibit exploration, you reduce exploration, and you are anticipating something to happen at a specific time. And then what happens after this thing happens? Now there is a big effect of inhibition after. There is some before and a bigger inhibition after, and then there is a release of inhibition. So do you think that the same explanation actually apply after the stimulus arrives, because now you are processing the stimulus and you don't need to explore anything, you are exploiting actually. So that's...
Right, I completely agree with it. This is 100% agree, yeah. I think yeah.
Okay.
No, yeah, you described it perfectly, just as I would.
So I just wanted to make a note about the role of microsaccades. So one idea is that they give a global synchronizing signal, because it's something that affects the whole brain, you would say. I mean, you get a visual signal that is very accurate in time, which is, we see it, for example, when you are listening to music, and actually your eyes become synchronized in a way with the music, with the thing you are attending. Okay, so another...
But this would also apply to large saccades, right?
True. But yeah, we do experiment when
there's a lot of advantages. There's good reasons why we move our eyes all the time. And but I still we're never convinced by any dissociations between small and large saccades.
All right, but then small or large, they do synchronize with the music in a way that amplifies the synchrony with the same thing you are trying to attend. So, okay, that's right. So I really liked another question, or I really liked your experiment with the salience, or the conceptualization actually of salience vs relevance.
Mm-hmm.
And we think that now we are thinking in a similar way, but that made things clearer to me. But in relation to autism, to severe autism, where we tend to go more for the salience and less for the relevance. And we are thinking how to measure this, how to quantify the and that's why I asked you if you can manipulate the ratio between salience and relevance by measuring the effect of the salience versus the relevance, or something to.
Yeah.
Like that.
And that's interesting point. I would love to talk more about this, because I think this is it's an interesting direction. And I don't have any input right now to give about this, but this is definitely an interesting step to go from here.
No other question.
No other questions?
So I'm sure people have more questions, but maybe people can write to Shlomit or to me if you're not feeling comfortable with asking them right now.
Feel free.
So again, Shlomit, it was a wonderful talk, beautifully presented, and really interesting results. We're very glad to have you in our seminar, and many thanks.
Thank you.
I want to know when we can have the third part.
Yes, maybe it was premature for me to crop it to stop.
I'd be happy.
Okay, maybe we'll invite you. We have a few more slots available this year, so maybe we'll invite you for another talk, for a complimentary.
Thank you.
For a complimentary talk. That would be wonderful. I also want to say that next week we're supposed to have Marcelo Rosa from Monash University. He'll be talking about visual system structure and organization. I invite everybody to join. It will be in the same, he's, I think, in Australia, but so it's going to be the same time this week on Tuesday. So many thanks, Shlomit.
Thank you.
Thank you.
And a big applaud.
Bye, Shlomit. Goodbye.
Bye.
Bye.
Bye, bye, bye.